Good morning everyone. Today's topic is differences between prokaryotic cell and eukaryotic cell. The word prokaryote and eukaryote was first coined by Sir Edward Chatton. So pro refers to before, karyote means nucleus. U refers to true, karyote means nucleus. Eukaryotic cells are the cells that have a completely developed nucleus, which is a true nucleus. These prokaryotic cells do not have a completely developed nucleus. Instead, they have a region called nucleoid where the genetic material is present. And this prokaryotic cell contains this nucleoid. No, it is considered as a stage that is present before the development of a complete nucleus. Hence, it is called prokaryote. So, the definition for prokaryote is that any unicellular organism that does not contain a membrane bound organelles or nucleus. So here we are referring only to unicellular organism, because only unicellular organism will not contain membrane bound organelles and nucleus inside them. And here any cell that contains nucleus and membrane bound organelles are called eukaryotic cell, because it includes both unicellular and multicellular organisms. So the examples for prokaryotic cells are bacteria and archaea. The singular form of bacteria is bacterium. The singular for archaea is archaeon. And the examples for eukaryotic cells are animal cell, fungi, plant and protist. And moving on to genetic material, the genetic material is present in the cytoplasm in a region called nucleoid. Nucleoid is not a different organelle, or it is not any separate space. It is just a place where the genetic material is present. So this is how a nucleoid will be present in a bacterial cell. And moving on to eukaryotic cell, the genetic material is present inside a structure called nucleus. This nucleus has four important components. The first is the external membrane that is surrounding the nucleus. It is called nuclear membrane. It is a double-walled membrane, and inside this nucleus we have a small spherical bodies called nucleolus. This nucleolus has rDNA, RNA polymerase, ribosomal proteins and RNA inside this. It is a site for, it is the site for transcription and the arrangement of ribosomal subunits. They do not have DNA or chromosomes inside them. And this chromatin network is a thread-like structure that is present inside this nucleus. So this is the one which holds the DNA, that is genetic material of the cell. And this last segment is a pore. Pore refers to hole-like structure. So these refers to small holes that are present in between the nuclear membrane. And the next point is about chromosomes. Chromosomes are single and circular in case of prokaryotic cell. In case of a eukaryotic cell, we have more than one chromosome. So it is several in number and it is a linear chromosome. It is not circular, it is just a linear one. And the last, so the next one is organism type. Here from this example itself we can understand that prokaryotic cell is present in unicellular organism, but in case of eukaryotic cell, it includes both multicellular and unicellular organism, but predominantly multicellular organisms are eukaryotic in nature. And the sixth point is cell size. The cell size of prokaryotic cell varies from 0.2 to 2 micrometer in diameter, whereas in case of eukaryotic cell, the size varies from 10 to 100 micrometer in diameter. And the seventh point is cell wall. The cell wall of prokaryotic cell are complex, and all prokaryotic cells will have cell wall. And the components of prokaryotic cell are, in case of gram-positive bacteria, the cell wall is made up of peptidoglycan, but in case of gram-negative bacteria, the cell wall is made up of peptidoglycan and lipopolysaccharide layer. In case of gram-positive archaebacteria, the cell wall is made up of pseudomurin, methanochondroitin and heteropolysaccharide layers. In case of gram-negative archaebacteria, the cell wall is made up of crystalline proteins and glycoprotein subunits. And moving on to the eukaryotic cell wall, all eukaryotic cells do not have cell wall. For example, animal cell lack cell wall, and the remaining eukaryotic cell which has cell wall, its composition will be very simple, and the components of eukaryotic cell wall are pectin, chitin in case of fungi, and cellulose in case of plant cell. And the next point is about plasma membrane. Plasma membrane is a layer that is present inside the cell wall, and it is a layer that encloses all the content of the bacterial or any other cell. In case of prokaryotic cell, this plasma membrane will not have any receptors. But the eukaryotic cell will have certain receptors in their plasma membrane, and these receptors are made up of carbohydrates and sterols. The function of this receptor is that it acts as a connection between the cell and its extracellular space. And the next point is about glycocalyx cover. This glycocalyx cover is nothing but an additional layer that is present outside the cell wall. It is different from capsule, and this glycocalyx cover is made up of polysaccharide and polypeptides, and this acts as a barrier between the cell and its surrounding. Some bacteria will have glycocalyx cover, whereas the eukaryotic cell that do not have cell wall will have glycocalyx cover. And the next point is about flagellum. Not all prokaryotic and eukaryotic cell will have flagellum. Some prokaryotic cell will have flagellum and some eukaryotic cell will have flagellum, and that's why I have written it as if present. Flagellum, if present in prokaryotic cell, it is simple and it is made up of flagellin protein. And this flagellum, if present in case of eukaryotic cell, it is complex and it is made up of microtubules. And these are hair-like appendages. Hair-like appendages are present both in prokaryotic cell and eukaryotic cell. Again, it is not a mandatory structure. Some cells will have these hair-like appendages, and if present these hair-like appendages in case of prokaryotic cell, they are called as pili and fimbriae. Pilus is singular, fimbria is singular. Pilus is larger than a fimbria. Pilus is less in number than a fimbria. And in case of eukaryotic cell, we have hair-like appendages called cilia. And the 12th point is about genome. Genome is the genetic material of a cell. The genetic material of prokaryotic cell is haploid in nature. It is represented by N, and which means they have only one copy of each gene. But in case of eukaryotic cell, it is diploid in nature. Di means 2. It is represented by 2N. So they have more than one copy of each gene. And the genome of prokaryotic cell is very compact, that is, they have very little repetitive DNA sequences. But in case of eukaryotic cell, they have a large amount of non-coding and repetitive DNA. And this genome is present in the cytoplasm in case of prokaryotic cell because they do not have a well-developed nucleus. And this genome is present in the nucleus in case of a eukaryotic cell. And the next point is about DNA transcription and translation. In case of a prokaryotic cell, both the transcription and translation will take place simultaneously in the cytoplasm. They both will take place in the same time in the cytoplasm itself. But in case of eukaryotic cell, the transcription will take place in the nucleus, whereas the translation process will take place in the cytoplasm of the cell. And the next point is about growth rate or generation time. The growth rate of prokaryotic cell is very fast, that is, it is faster comparing eukaryotic cell. And the growth rate of eukaryotic cell is slower comparing a prokaryotic cell. And the next point is about ribosomes. Ribosomes are the site for protein synthesis. They link the amino acids together to form a polypeptide chain. In case of prokaryote, they have a small ribosome that is 70S in nature. This 70S ribosome has two subunits, 50S large subunit and 30S small subunit. This 50S large subunit contains 23S rRNA, 5S rRNA and 34 proteins. This 30S small subunit contains 16S rRNA and 21 proteins inside it. In case of an eukaryotic ribosome, it has two subunits. One is 60S large subunit, the other is 40S small subunit. This large 60S subunit contains 25S rRNA, 5.S rRNA, 5S rRNA and 49 R proteins. This R refers to ribosomal proteins, ribosomal RNA, and this small 40S subunit contains 18S rRNA and 33 R proteins inside it. This S refers to Swedberg unit. It is named after the scientist Sir Theodore Swedberg who discovered it. This Swedberg unit refers to sedimentation coefficient of a particle in an ultracentrifuge machine. So, the next point is about DNA replication. The DNA replication is different in case of prokaryote and in case of an eukaryote. For example, the origin of replication is the point where the replication starts. In case of bacteria, we have single origin of replication, but in case of archaea, we have some will have single, others will have multiple origin of replication. Since archaea has both the characteristics of bacteria and eukaryotes, this is possible in case of archaea. Whereas eukaryotes will have multiple origin of replication. And the second point is about number of base pairs per second. In case of prokaryote, the replication process is very rapid, that is 2000 base pairs will be added per second, but in case of eukaryote the process is very slow, 100 base pairs is added per second. And the third point is about DNA gyrase. This ase is a suffix which denotes that this word indicates an enzyme name. Here gyrase is an enzyme that this gyrase is present in case of prokaryotes, but in case of eukaryotes they do not have gyrase enzyme. This gyrase enzyme will catalyze a process called negative supercoiling. So this negative supercoiling will be done in case of eukaryote by some other mechanism, and hence this gyrase is not present in case of eukaryotes. And the 17th point is about histones. Histones are proteins that are present in the nucleus which gives structural support to the chromosome. Histones are absent in case of prokaryotes, but they are present in case of eukaryotes. The DNA is wound around the histone in such a way they help in the formation of chromosome structure. And the last point is about reproduction. Reproduction is a process by which an organism produces its offspring. In case of prokaryotes, we have asexual reproduction which involves only one parent cell. Bacteria reproduces by binary fission, archaea reproduces by binary fission, fragmentation and budding. In case of prokaryotic cell, we do not have sexual reproduction, but there is a process called conjugation, through which we have exchange of genetic information between two organisms. The one organism that gives the DNA is called donor, the other bacteria that receives the DNA is called recipient. In this process we have the involvement of two organisms, but it is not a complete sexual reproduction because here we have the participation or the role of one bacteria, the other remains inactive in this process. And eukaryotic cell reproduces by sexual method where the fusion of gametes will take place. There are some eukaryotic cell that reproduce by asexual method also. Thank you so much for listening to the entire video. Make sure you subscribe to my channel